Hello, everyone. Today we are going to talk about viruses. So, the first thing we need to know is what exactly is a virus? We've heard of viruses. We're in the middle of a pandemic because of a virus. So, what exactly is a virus? That's really what we're going to start with today. So, a virus is very small, right? So, in terms of scientific history, they're a fairly recent discovery because they are much, much smaller than a bacteria. So here we see the comparison. We have a red blood cell and the comparison inside of the bacteria to that. And in comparison to a bacteria, which is very small, viruses are much smaller, right? Even large viruses like a bacteriophage or a smallpox virus is much bigger than some of these other viruses. So they're very tiny. So is a virus a living thing? Well, the answer is not really, right? It doesn't have all of the components of a living thing. So viruses are sort of alive, but not really. They have some characteristics of living organisms, but they're not considered living. So how are they like living organisms? Well, they're like living organisms because they have a genome made either of DNA or RNA. So they have genetic material. Viruses can mutate, and therefore viruses can evolve. They can replicate, and they can interact with other organisms. So it sounds like they are alive, but in fact, they are not like other organisms because they are actually derived from the cells of other living organisms. So they can't derive themselves from their own equipment. They need the equipment of a host cell. They have to reproduce using a host cell. They don't have their own metabolism, so the energy that they use has to come from their host cell. And they don't respond to stimuli, so they are not going to be changing. They really sort of do one thing, and that's to replicate and infect. So a virus is referred to as an obligate intracellular parasite. So it's a parasite because it has to rely on other things in order to exist. Viruses can only replicate within a host cell. So a virus structure is very simple. There is nucleic acid on the inside, either DNA or RNA, and then there is a capsid. This is like a protein coat surrounding it. And then there are recognition spikes. So these are proteins that protrude into the environment, and this is going to help them get inside their host cell. So here we see that again. We have the nucleic acid. We have the protein capsid. Some viruses have what's called a lipid envelope. So this lipid envelope may be derived from something they infect. And these are some of the spike projections that are from on the outside of the virus. So this virus has an envelope, but they don't all have an envelope. So viruses are very diverse, and we'll find out why later. So you can see here they have a variety of different shapes, of different sizes. Here's the one that infects bacteria, and here's the influenza, adenoviruses, a plant virus. So viruses can infect plants and animals and bacteria and all sorts of living things. So classifying viruses is very difficult. And the reason why is because they are tiny. Their genomes are also very tiny. They're very short. And their tiny genome size, the fact that it's so short means that their mutation rate is going to be rapid, right? Because any change to their genome, because their genome is so small, any change is going to have a bigger effect, right? Like imagine you change one word in a short sentence. That's probably going to have a big effect on the sentence. But if you change one word in an entire book, it's not going to have quite the same effect. So because their genome is so small, they mutate pretty quickly. We don't know of any fossils of viruses, so we don't really have too many clues into their origins, and they're very diverse, so they're hard to classify. Some of the ways that we can classify viruses are based on whether they have RNA or DNA as their genetic material, and whether they are double-stranded or single-stranded. So let's first talk about RNA viruses. RNA viruses are viruses that have RNA as their genetic material. And they probably represent escaped components of cellular life that now evolve independently of their host. So it could be that somehow these pieces of RNA escaped from a very early cell and became a separate entity. Scientists speculate that single-stranded RNA genomes may have been common in the distant past before DNA became the primary heredity molecule. So we know that RNA is older, so it could be that RNA genomes and viruses come from that time when RNA was the main genetic material. A self-replicating RNA polymerase gene that began to replicate independently of the cellular genome could conceivably acquire protein coding genes through recombination with its host DNA. So if one or more of these viruses were to foster development of a protein coat, the virus might be able to then survive outside of the host. So if they could have their separate genome, and then if they could code for their own protein coat, it's possible that the genome and the protein coat could go outside of the host and survive on its own and infect another host. And that's how we might have ended up with a virus. So single-stranded RNA viruses, there's really two different types. In one case, the single-stranded RNA virus, the RNA that's present in the virus is a complement of mRNA. So basically, they have to copy the RNA to make the mRNA, and the mRNA will then code for the protein coat. So in this type of RNA virus, we need to synthesize mRNA with RNA polymerase. So we need an RNA polymerase to basically read the RNA to make more RNA, right? The complementary sequence to that RNA. And once we have that mRNA, then we can make more protein coats. Other types of single-stranded RNA viruses are already set for translation. They don't need to replicate, so they don't need an RNA polymerase to make a complementary strand. The strand they have present there is already good enough. RNA retroviruses like HIV are an interesting case of virus because they have RNA on the inside, but a virus like this, a retrovirus like HIV, is going to do something very interesting. So there is RNA inside the virus. So we've got the virus infects the host cell, the RNA goes in, and then a particular enzyme that's right over here called reverse transcriptase. So think about that. What is transcription? Transcription is taking DNA and turning it into RNA. So reverse transcriptase is going to be taking RNA and turning it into DNA. So reverse transcriptase makes DNA out of the virus's RNA. And then what that DNA is going to do is it's going to hide in the host genome. So actually in the host cell's DNA, that HIV DNA is going to slip in there and hide. And the cell will not have any idea that it has been infected. So it's like hiding there. And then we'll talk about this more later, is that at some point that DNA that's hiding in the host genome can come out and can express itself. And then once that DNA is expressed, it can make more viruses, and then those viruses can go out and infect other cells. So this particular retrovirus, it's an RNA virus that can hide. So you have RNA becoming DNA and waiting and hiding. All right, double-stranded RNA viruses could have evolved from single-stranded RNA, or perhaps the opposite is true. So again, this is another way of classifying viruses. And then we have DNA viruses. Some of the DNA viruses could be single-stranded or double-stranded. Either way, they're going to work in a very similar way. So here's the viral genome. It goes into the cell. The cell is going to replicate the other strand. And then this strand is going to potentially make RNA that can then make proteins. and those proteins can then go on to make more viruses. And the same thing with double-stranded DNA. The process is a little bit simpler. This double-stranded DNA is going to use the host cell's own RNA polymerase to make RNA, and then this RNA can be translated to make proteins, and those proteins can come together to make more viruses. DNA viruses, where did they come from? It's possible that these viruses are highly reduced parasites that have lost their cellular structure as well as their ability to survive freely. So they have to exist in a host or in their capsid, which they can't really do anything in their capsid. So it's possible this represents some sort of reduced parasite, or it may have emerged from some very simple organism long, long ago and mutated from there. Some viruses have a larger than average genome that can encode for a lot of proteins, and this genome is similar in size to many parasitic bacteria. So that's a little bit of evidence, not that much, but it's a little bit of evidence that that could be a possible explanation. It appears that because we have this huge diversity of viruses, it's because all these different viruses have multiple origins. So it's not that there was one initial virus and it differentiated into all these different viruses. It could be that viruses have evolved many times throughout history. It may be best to think about viruses as spin-offs from the various branches of the tree of life. So it could be that perhaps viruses that infect mammals are spin-offs from some early mammal. Viruses that affect plants then would have maybe been spun off from a plant or a plant-like protist. So there are a number of hypotheses for how these viruses emerged. And here we can see the huge diversity. Influenza, HIV, both have an envelope and they have those spikes, a bacteriophage, a plant virus, Ebola, cowpox, the common cold. So we have all kinds of different viruses and they're quite different from one another. Okay, so how do viruses reproduce and what do they do once they infect a host? There are two different life cycles for a virus. There is the lysogenic cycle and the lytic cycle. So both of these are going to be two slightly different methods of reproduction. So I think we have a picture here. Okay. So here we have, let's start at the top in the middle. A bacteriophage will infect a bacteria. All right. So when it does that, it injects its DNA. Or DNA or RNA, whatever its genetic material is. Now, if this virus enters the lysogenic cycle, it's going to go to the right over here. And what that means is kind of like we talked about with HIV, the genetic material that the virus injected into the cell is going to hide in the host DNA. We refer to that as a prophage. And the cell has no idea it's been infected. So the cell reproduces, and because all DNA is pretty much the same, it accidentally reproduces the viral DNA that's hiding there. And so this reproduction can happen again and again and again and again. At some point, for some reason, sometimes the viral DNA will exit the host DNA. Why that happens, we're not really sure. It could be some external or environmental factor that triggers it. And when that happens, we move from this lysogenic cycle of hiding to the lytic cycle, which is viral reproduction. So what happens when the DNA comes out? Well, when the DNA comes out, basically, this DNA is then going to be expressed, right? This DNA is going to be transcribed and translated, and proteins are going to be made, and those proteins are going to cut up the host cell DNA. So the host cell is done at this point. Then this DNA is going to be expressed, and the capsids are going to be produced. So we're basically using the host cell's own machinery to make more proteins that can be assembled into more viruses, and those viruses are going to explode out of the host cell the host cell is done, the host cell's been killed, and now they can infect other cells. So the whole thing starts again. Some viruses go right into the lysogenic cycle, and they can wait there for a long time, even years. Some cells go right into the lytic cycle, and they start infecting right away. So two different cycles. All right. We're going to stop there for now. So if you have any questions, let me know.